del Auditorio Alfonso Escobar del Instituto de Investigaciones Biomédicas. Este auditorio tiene una capacidad para 233 personas. Cuenta con una salida de al frente, en su costado izquierdo, 2 salidas en la parte posterior y una área de reunión al salir del auditorio. Es que usted espere en su lugar hasta que finalice. Posteriormente, salir en forma ordenada hacia el punto de reunión para recibir indicaciones de los brigadistas. El Instituto de Investigaciones Biomédicas de la Universidad Nacional Autónoma de México agradece su atención y les da la más cordial bienvenida.
Hola, buenas tardes a todos. Muchas gracias por estar. Le damos la bienvenida también a nuestras sedes foráneas, como Tlaxcala y Veracruz, que están conectados. El día de hoy tenemos una invitada especial, la doctora Meytal Landau. Ella estudió su licenciatura en farmacia en la Universidad Hebrea de Jerusalén. Posteriormente realizó una maestría en neurobiología y un doctorado en bioquímica estructural en la Universidad de Tel Aviv. Realizó su postdoctorado con el profesor David Eisenberg en la Universidad de California, UCLA en Los Ángeles. Y la doctora Landau ha publicado más de 60 artículos que han sido citados más de 3.900 veces y tiene un índice H de 27. Ha publicado en revistas de alto impacto como Science, Nature Communication, Nature Structural and Molecular Biology, PNAS, entre otras. La doctora ha obtenido diferentes grants de Alemania, Estados Unidos e Israel. Forma parte de los laboratorios europeos de biología molecular como profesor asociado, visitante y miembro asociado en los laboratorios CSB y DESI de Hamburgo en Alemania. Actualmente también es profesor asociado, líder de grupo en Technion Institute Israel, que es el Instituto Israelí de Tecnología en Haifa, en Israel. Su línea de investigación la define como: nuestro objetivo a largo plazo es desentrañar la estructura y los principios mecánicos de amyloides específicos, sus funciones en la fisiología y en la salud y la enfermedad humanas. En última instancia, esta comprensión conducirá al dominio de la biología amyloide y ofrecerá nuevas vías para la ciencia, la industria y la medicina de las proteínas. Doctora Landau, welcome. Shalom Alechem Tavo Beit Sheli.
Great to be here in a different part of the world. I'm very excited about that. And I'm also excited to tell you about my amyloids. I can tell, yeah? I feel strange. I can tell my amyloids that they travel across the pond, like across the, yeah, I'm tall. Yes, the ones I landed in Mexico, I felt super tall. It's when I'm traveling to Germany, I feel intimidated, and in Israel, I feel okay. So it's really nice for me to travel and to see part of the world, and I was very impressed with the size. I don't know how you hold university in this scale of students and how. You handle so many people, and at the same time, it's very impressive. We are not as big as you, like the institute that I'm coming from, maybe 10,000 people, including all the students and everything. So it's a different scale. Okay, but I'm happy to introduce my amyloids. They are proteins that I feel emotionally attached to very much. So I want at the end of my talk that you will also like them as much as I do. I think they're a very interesting protein. So I work on amyloid fibrils. Maybe most of you know amyloids from associated with neurodegeneration and amyloids and amyloidosis. But the amyloids that I'm interested are involved in infectious diseases. They serve as key virulence determinants in microbes. they're killing host cells or they're scaffolding biofilm-resistant barriers. Of course, they pose a major threat for both public health and the environment. And it's a relatively unexplored field. And our work are in the past 5 years provided the first structures of bacterial amyloids and revealing similarities and surprising differences compared to human amyloids, and I will tell you about them. And the work on the microbial amyloids led us, and I will show you how, to discover a new set of amyloids of antimicrobial peptides that are being secreted by organisms from all kingdoms of life. So now it's time for us to understand what is the connection, what are the differences, how they relate to each other, and what are the mechanisms that they use for their activity. So, but I want to go back to introduce a little bit about amyloids, because they're really being explored for many years now in relation to neurodegeneration, Alzheimer's and Parkinson and ALS and Huntington, and of course, prion diseases. And maybe the most famous ones is that we know that they form plaques in the brain. So nobody really knows what happens in Alzheimer's. We don't understand the etiology of the disease. We don't really know what causes the toxicity to the neurons and initiating the progress. But we know that in Alzheimer's patients, we have plaques of proteins in the brains. We know that the disease starts maybe 30 years before we have the symptoms of Alzheimer's and Parkinson. So we know the endpoint of protein aggregation in the brain. We don't know the start. And I think one of the novel discovery was by Stanley Prushner tried to push that for years, and the community resisted, because he claimed that amyloids, especially prions, which are a type of amyloids, they can be infectious. So all of these diseases like the mad cow disease, I don't know if you remember this from the 90s, in sheep it was called scrapie, in cows they called it a mad cow disease. And then in the 90s, there were indications that humans can be infected by consuming contaminated meats that are coming from these cows with prions. So it means it's a transmissible disease. You eat meat and you get a disease. But in contrast to all of the infectious diseases that we know that are being transmitted by microorganisms, in this case, it's transmitted by protein. This is why what Stanley Prushner got his Nobel Prize for understanding that transmissibility can be by protein. And if you think about it, this is pretty crazy, because we are evolved to eat proteins, and they're being digested in the acid in our gut, and then all the proteolysis processes, like in the stomach, in the gut. So how come a protein that we eat can survive all this way and reach the brain and template a disease? So it's a question that people didn't know how to answer for years. And from the point of structural biology, which is my field, we said, okay, let's explore the structure of a protein that can be super, super, super stable. And this happened before I even reached amyloid. It's the first structure of fragments of amyloid that, so amyloids can be huge proteins. And all of the structural work in the first 20 years, first 10 years or 15 years was done on only the backbone of the amyloid. So what formed the amyloid fibril. And the first structures were determined by David Eisenberg's lab where I did my postdoc, but the first one was before I joined, it was 2005. And they revealed this motif that you can see molecules that are stuck perpendicular to the fibril axis forming mated sheets. So you can imagine that you have beta sheets with all the hydrogen bonds along the sheets. And then you have two sheets mated together and the side chains facing inside the sheets, and they interdigitate. And you can imagine it as a steric zipper, as a zipper, you know, like a zipper in the pants. You can imagine it as a zipper. So you have hundreds of thousands of layers over the beta sheets that form this zipper. So the Eisenberg lab called it a steric zipper, and the more known term from amyloid is cross beta. So this is how amyloids were defined until very recently, as cross beta forming proteins. So they have this super stable structure, and this structure also illuminated the stability of the amyloid fibrils and their self-seeding characteristic. It means that you can imagine this beta sheet growing, and then it meets, you have a seed of this fibril, and then you see a prion protein in the brain. Even if the prion is coming from a cow, it's similar enough to the prion in human to just recruit the protein and add it to the fibril. So the fibril is growing by recruiting more and more protein that just assume this configuration of the beta sheet. This is the basis of amyloid formation. And since then, the Eisenberg lab determined more than 200 structures of amyloids and prions, different proteins, and they all show the same properties of the beta sheets. And over the years, especially with the resolution revolution of cryogenic electron microscopy, CryoEM, so more structures were determined, including directly from the brain of Alzheimer's, or the brain of Parkinson, or the heart of people with amyloidosis. So we have more and more structures, and they look very diverse, like they look different. But if you look carefully, all of these amyloids are anchored in extended mated beta sheets that run along the fibril axis. So the molecules are stuck perpendicular to the fibril axis. This is what defines an amyloid. And this was what was known until recently. But these are all human amyloids. And now I want to, one of them is actually from Drosophila. So amyloids are involved in long-lasting memory in flies. So it's one of these structures is actually from Drosophila. But all of these structures that were determined are coming from humans or a different type of eukaryotes. But what about microbes? So in 2002, Matt Chapman found that biofilm, the proteins that are associated with biofilm of E. coli, also have amyloid-like features. But there was no structural information. When I started this work about 7 years ago, there was no structural information, nothing. The field was completely empty. What happens if bacteria and microbes can form amyloids? So people knew, like they had characterized protein here, protein there, and they saw by biophysical measurements that it formed the fibrils. They even understood that it has beta-rich structures, but there was no high-resolution structural characterization. And we started with this work. You cannot work structurally. Well, very recently, it's possible, but until very recently, with advances in cryogenic electron microscopy, it was impossible. So the only way is to use this Eisenberg approach and to look for the spine of the amyloids and determine their structures with X-ray crystallography. It's also a relatively unique field because amyloids are very different compared to globular proteins. But we solved structures from different microbes, and every structure that we solved showed that it forms this steric zipper cross beta structure. So on one hand, as a structural biologist, it wasn't very exciting because I solved structures in my own lab and it was fun, but they looked exactly as the Eisenberg structures. So structurally wise, they were not interesting, but it was the first time that this specific configuration was seen in bacteria. So from this structure, we know that steric zippers can go all the way from prokaryotes to eukaryotes. And we thought about what of the implication. If biofilms are really similar to human amyloids, what does it mean? So first of all, it can mean bad things. It can mean that if cows' proteins can template the human amyloids, is it far-fetched to think that amyloids from microbes can do something similar? So it's called maybe the gut-brain axis, the relationship between microbes and propensity to Alzheimer's, and they're accumulating evidence today that there is a connection. For example, if you take a mice model of Alzheimer's and you inject to the stomach the E. coli that expresses these amyloids, you have more severe symptoms of the Alzheimer's. You can find chlamydia in plaques of brains with Alzheimer's. You can find herpes viruses in plaques of brains of Alzheimer's. You know that the microbiome of Alzheimer and Parkinson patients are different than the microbiome of healthy people. So there are all this causality, it's only in the causality level, but there is definitely a relationship between microbes and neurodegeneration. And we don't really know what is the relationship, but some of the hypothesis is that microbes do change the propensity for Alzheimer's, because except from particular cases of genetic related Alzheimer's, most Alzheimer's are sporadic. Like we don't know what they are happening. So maybe it's our microbiome. We know that our microbiome is responsible for a lot of things. So maybe also this propensity too. So this was one aspect, but the other aspect was good. The other aspect said, okay, if they have similar structures, maybe they can be inhibited by the same compounds. And there are so many compounds that were already in clinical trials for Alzheimer's. They were tested for safety, for toxicity, everything. So maybe we can take some of the compounds and see if they work on biofilm. And this is what we did. We collaborated with a group in Germany, and we took compounds that they developed and they were already in clinical trial. And we found two compounds that inhibited the biofilm-associated amyloid from E. coli and Salmonella and reduced the biofilm biomass of Salmonella. So think how cool this is, that you can repropose drugs coming from Alzheimer's to serve as anti-biofilm compounds. And until now, we don't have any compounds in the industry or the clinic that can really treat biofilms. So it doesn't disassemble biofilm, but it prevents biofilm formation. So we are more thinking about coating materials for everything. It can be pipes, it can be medical devices, it can be food packages that will prevent biofilm formation based on compounds that were developed for Alzheimer's. And for Alzheimer's, they don't work not because they don't inhibit the protein. They do inhibit the protein, but probably way, way, way too late. So Alzheimer's, you know, once we already have phenotypes, we have plaques in the brains that are no reversible. And I want to share with you that you can appreciate these plaques. So we work on these proteins in the lab. They are crazy proteins. We call them lab killers because they're so strong, they're so sticky, you cannot put them. If you use a cuvette, it has to be disposable. When we purify them, you cannot use fancy FPLC machines. You have to use only disposable pipes, because once they are formed, you really cannot eradicate them. We use SDS page to analyze inhibitors because they are happily growing in SDS buffer at 95 degrees. They keep on growing in this buffer. It's not just killing them, and they don't run on the SDS page. So the way to monitor some of these inhibitors is to see that we do manage to run them on gels. Like it's that crazy and simple at the same time. And one of the more crazy amyloids is from the Pseudomonas aeruginosa. It's Nimrod is working on this. So this is an unpublished work, but we characterize the amyloids from Pseudomonas. So one of the compounds from Alzheimer's did manage to inhibit one of the amyloids from Pseudomonas, but only slightly inhibiting. It's not very good as it is for the E. coli and Salmonella. And Nimrod, he works on these proteins for a couple of years now, or 3 years now, and he always plays with them and to see how extreme he can go. So these proteins survive 100% formic acid, and he put them in the autoclave, and they're still there. So if you have Pseudomonas infection, maybe in autoclaving your tools, you are getting rid of your bacteria, but you are not getting rid of the amyloid fibrils in your biofilm. So they're really crazy proteins. And when you see something like this, you understand the basic of prion diseases. If they're so super stable against temperature, chemical denaturants, proteases. So it's not that surprising that we can eat contaminated meat and it survives, right? So it explains the stability of prion protein and the basis of amyloid transmissibility. So Eisenberg called that a protein encoded inheritance, because you can actually transmit the amyloids to the next generation, not by genetic means. So, so this was on the biofilm, and now I want to tell you about my favorite form of amyloids, and they are starting with virulence factors. So there are amyloids that are actually killing our neutrophils, our immune system. They're being secreted by microbes, so they are very, very virulent. And we worked on a bacterial protein from Staphylococcus aureus. It's called PSM alpha 3. It's a peptide. It's a 22 residue peptide. This is how it's being secreted. And this PSM family of Staph aureus, so the most virulent strains of Staph aureus, they 60% of the protein mass they secrete outside, it's PSM. So they are very important for virulence. And there were indications that they form amyloids too. And we saw by electron microscopy that they form these nice fibrils. They bind the amyloid indicator dye, and we got crystals of them. And I was super happy that maybe we can determine the structure of this amyloid. But I was also surprised because I told you in the Eisenberg lab, we only worked on the core of the amyloid. It was never longer than 8 residues. Anything that is longer than 8 residues did not form crystals. So, and here it was the full-length peptide, the way it's being secreted, and it's 22 amino acids. So I was surprised, why does it form crystals? And they look exactly the same as the amyloid. You can see that they look like needles. This is how amyloid crystals look like, because they are actually between fibrils and crystals. And I thought that something is wrong, and I sent the students to recheck, and we sent it to mass spec, but there were crystal of this peptide, and it took us maybe 7 trips to the synchrotron. It was a different synchrotron in Europe, one in the US, until we got the data. So it was about a year until we managed to solve the structure. But when we solved the structure, I understood why I was fighting for it. Like when I didn't want to let this go. And I also understood why it forms the crystal, because it formed amyloid exactly like the other amyloids, but it was formed of alpha helices and not beta sheets. So we called it a cross alpha. So you can appreciate that the quaternary structure of the cross alpha is exactly the same as the cross beta. They are formed from molecule stacked perpendicular to the fibril axis, forming mated sheets. Exactly the same, but the secondary structure is surprisingly alpha helical and not beta sheets. And this is how it managed to crystallize, because the helical was ordered along all the length of the peptide, in contrast to beta strands that cannot go that long. So it was a cool structure, and it was exciting in terms of finding a new form of amyloid. It was exciting from the protein science level, because nobody, like helical sheets, it's not something that you see every day, right? It's not something that you even expect. But okay, so besides the fact that it's a cool structure, then especially the editor asked us, okay, you want to go to science, you have to also tell us if it's doing something cool, not just looking cool. So we worked on that for several months, and we hypothesized, and then we reinforced that the cross-alpha fibrillation is imperative for the toxicity of these peptides against human cells. And our model was co-aggregation with membrane lipids. So the peptides are interacting, what we think, they're interacting with the membrane, they are forming the fibrils on the membrane. Actually, the lipids in the membrane is accelerating fibril formation. And the cross alpha structure, it has a hydrophobic core because it's made out of amphipathic helices. So the core of the fibril is hydrophobic. So it's taking the lipid into the core. So it's basically tearing the membrane apart. In Israel, I give the example of pita bread, but here I can use tortilla. But imagine that you rip down the tortilla. It's like you take small pieces. It's like you take small pieces of the membrane and you recruit them to your fibril. So this is our model of how this works. Recently, another group from the US, Ed Engelman and Vincent Conticello, they solved the CryoEM structure of the same peptide, and it forms these beautiful nanotubes. So the basic unit of this nanotube was exactly what we saw in the crystal structure of mated sheet, but CryoEM can give you something that crystallography can't. Like crystallography is bound by symmetry. So everything looks the same, like across the 3-dimension, but CryoEM, which is not bound by symmetry, can tell you the larger supramolecular assembly. So in these specific conditions that they tested, it forms these beautiful, beautiful nanotubes. And I'm more than I'm impressed by CryoEM, I'm impressed by Staphylococcus aureus, because this family of PSM that it secretes, I will not show you all the data, but I will tell you that there are just 4 peptides, and 2 of them, they form the cross beta structure and are involved in biofilm. And one of them is forming the cross alpha and is killing our immune cells. And it was shown that they're being chopped in vivo to active derivatives. So there are proteases from the staph aureus and the host that are chopping this peptide into active derivatives. We saw the structure of one of them. It's also a new amyloid form, and they're killing microbes. So just by these 3 peptides that staph aureus secretes, you form biofilm, you stabilize, you don't form, you stabilize the biofilm, you kill the host immune cells, and you're killing competing bacteria. So really, it's an amazing, I think, pathogen. And I have a personal relationship with this pathogen because I couldn't walk for 2 years after it infected my hips. So for me, it's time for revenge. I didn't do it on purpose, but it's really time for revenge. And the time of revenge for me is now that we know the structure is to find inhibitors for the fibrillation. And I can tell you that up to now, we didn't find any. So it's really hard. So they are not inhibited by the same ones that are for the Alzheimer's, because they are cross alphas and not cross beta, even though they bind the same dyes. So up to now, we didn't find good enough inhibitors. But this is, of course, a mission that we are going to pursue in the following years. And okay, so we want to inhibit them, right? But also the question is, if the cross alpha structure is it a one-time thing? Is it the only example in nature that you see this cross alpha structure? And you can guess that not, because evolution it doesn't tend to do one thing once, right? It's usually a feature. And then we thought, where can we find more cross alpha? How do we even look for them? And the answer came from antimicrobials. So when after we solved this structure, there was papers coming out from Moyer and Tanzi in the US that amyloids that are associated with Alzheimer's and Parkinson, they have antimicrobial activity. So they had a theory that maybe the physiological role of amyloid beta alpha-synuclein is to protect our brain from pathogens, because the brain doesn't have the same immune system as is in the periphery, right? So it has to have its own immune system, and amyloid beta easily kills different pathogens. So there started to be this correlation between antimicrobial activity and amyloid formation. And we thought, okay, maybe the answer is there. And we just started to look in the literature for antimicrobial peptides that form fibrils, that were reported to form fibrils. And it led us to discover this antimicrobial peptide secreted by an Australian toadlet. I will call it an amphibian or a frog. So it's a small toad, basically. And it's secreted on the skin. And you want to think, okay, like what are these properties of the amphibian needs? So first of all, we managed to solve the crystal structure, and we discovered another cross alpha structure, another cross alpha amyloid. But then I was super surprised that the biophysical measurement showed that there is a secondary structure transition from cross beta to cross alpha that is induced by membrane lipids. So this peptide can have 2 different secondary structures. And recently we managed in my group in Germany to determine the CryoEM structure of the same sequence. It's exact same sequence, no differences. And we got a cross beta structure. It's a rather unique one, but it's a typical cross beta. And then you ask yourself, why does this amphibian need a fibril that has 2 different structures? And what we think, like the model that we are now trying to check, is that, amphibian, they're sitting in the sun, they're jumping to the water, they really change conditions. So if they have antimicrobial protection, you really need to have it very, very sturdy, very sticky, that it will not dissolve in the water, it will not shed off the skin. And what's better than something sticky and stable than a cross-beta amyloid? So we think that the cross-beta amyloid is actually the non-toxic form that is also not attacking the bacterial cells. And this is what people think about Alzheimer's, that once you see these cross-beta fibrils, it's the end point. It's not the toxic species. And then once you encounter a bacteria, the lipid in the membrane of the bacteria is inducing the shift for cross alpha, which is attacking the cells. So it's basically the membrane of a specific bacteria that decides who attacks it and who doesn't. So the membranes is inducing the conformation that is actually attacking them. And I think it's relevant for all type of these amyloids. So if we go back, I'm teaching biochemistry to undergrads, and in the last few years, I feel like I'm a liar, that I'm teaching them all these dogmas, of one sequence, one structure, everything is globular, we are teaching them all these kind of things, and it's obviously not true. So I think it's a unique example that this peptides from amphibian of a sequence that can have two different secondary structure in the fibril form. Moreover, that was determined at high resolution. But if you think about it, it makes a lot of sense, because you want a stability and you want regulation. You want a signal for activation. So for me, it makes a lot of sense, and it's nice that it breaks the dogma. So the next question is how prevalent how cross alpha and the secondary structure switch are. So we devised a computational platform, very, very basic, just by some intuition about the biophysical features that you need for this cross alpha and the switch. And by now, we published only 14, but we now have dozens of antimicrobial and cytotoxic peptides that form cross alpha and cross beta amyloid. Some have the ability to switch. We see really unique properties. The antimicrobial tend to switch. The toxic ones, for example, from spiders, from hornets, from wasps, they don't switch. So it's really organized. It seems like we are only at the beginning of it, but it seems that it's related to the function somehow. And I think that the ones that switch, we didn't put organism in any of our search. We never put criteria of organism, but almost all of the switchers are coming from marine creatures or amphibians. And if you think about it, it makes sense in terms of stability, because marine creatures, they live, they have the sheer force of the water, you have the different temperature. So you want your antimicrobial peptide to be super stable. So these seems to be the switchers. And we managed to solve the CryoEM structure of one of these newly identified peptides, and it revealed this new conformation of cross beta with kinked beta sheets. And Eisenberg suggested previously, based on segments from human functional amyloids, that this kinked beta sheets is actually a signature for reversible fibril formation. Because if you have the normal extended beta sheets, they are super stable. But if you have this kink, it reduces the stability and gives you the ability to form and disassemble. And for something that is functional, you want this reversibility. So, we are starting to understand basic features, but still every new structure reveals morphologies that are surprising and are unpredictable by any current tool, including AI. So we are very proud that AlphaFold 2 cannot predict our amyloid structures. So, it's a buzz, and like, do you even need structural biologists now with AlphaFold? But, if a structural biologist working on amyloids, we are definitely needed, because AlphaFold cannot predict us. So, and you can see why, because if you have a sequence with two different secondary structure, obviously it's not easy, and you don't have representative of that in the structural databases. So you know, we have to build so many of them for AI programs to pick up on the unique chemistry of their assembly. So and this was an overview of what we discovered, and I want to share with you a little bit how we think about future plans, how we want to approach it. So most of what we do is structure-function relationship, and then we go to more to the molecular mechanism, how they really kill the cells. And at the same time, we expect to find converging and differentiating features. So you have amyloids from biofilm, antimicrobial, virulent. What is the similarities? What are the differences? And my passion eventually is to find these rules that can lead to amyloid prediction, classification, design. So I want to reach a point, maybe in my next talk, that you can give me a sequence and I will tell you if it forms an amyloid and what function it serves. So this is the idea. And how do we pursue, for example, structure-function relationship? So you have to bear in mind that these self-assembling proteins are fundamentally different compared to globular proteins. So it makes structure-function studies really complicated. So what is the role of fibrillation in the different activity? How is the fibril morphology encoding toxicity against specific cells or specific bacteria or scaffolding biofilm? So how do we correlate all of this information? And what is the role of the secondary structure switch? When it's playing out and when it's not? And what is the thermochemical and mechanical stability of these fibrils and how it's related to reversibility and function? So I showed you an extreme cases of autoclaving them, but the antimicrobial ones are not like that. Antimicrobial ones are not as stable as the biofilm ones. And I think part of the reason is that once you form a biofilm, it's there, it sticks. Like if the bacteria wants to move, it needs to cut itself from a biofilm and move away, but the biofilm stays. But with the antimicrobial peptides, you always want them to go back and forth. You don't want them to attack all the time. So they have to have some more features that allow this reversibility. And of course, we want to determine the high resolution structure, which is the most difficult part. But with every structure, we reveal new morphologies that allow us to generate new hypotheses. So it's important that we keep on going with the structure, although I can tell you that it takes many, many, many months of experienced people to determine each one of these structures, if it's even possible. It was difficult with X-ray crystallography, and it's very difficult, again, with CryoEM. And then we are testing doing all these studies with mutations and truncations and varying conditions because we want the environment to be very relevant to the physiological environment of each of these peptides. So you have to make your conditions for every system that fits its own, you know, if it's skin of a frog or something like that. So the structure-function relationship we conduct on several systems, and the parts that we work on is the antimicrobials, the amphibian ones, but they come from everywhere. Virulence factor from Staphylococcus aureus mainly, and the biofilm ones that we are now focusing on the Pseudomonas because they're super interesting. And then we choose a few of the toxic peptides, and then we go in depth into the molecular mechanism. But molecular mechanisms are super difficult. Why? Because you have to bear in mind that the self-assembling systems, they change while they're carrying out their function because they're affected by the membrane lipids of the cell they attack. Imagine to study mechanisms of something that changes during activity, like how difficult this is. Like we think about, okay, enzymes, but enzyme, okay, they open their mouth and close their mouth. But here we are talking about self-assembly and changing secondary structures. How do you do this? So for a profound mechanistic understanding, it necessitates visualization of the toxic process at high resolution. So our approach is to do it stepwise. First, we want to solve the structures of the fibrils in the presence of the lipids or the other molecules that they interact with. So we know what's the membrane interacting form. With AFM and nano-IR, it's a biophysical method that allows you to look on the secondary structure of single molecules. So look on the secondary structure of the fibrils that are contacting the cells versus the ones that are not. And the high-hanging fruit is to use a technology that is emerging, and it's a cryogenic correlative light and electron microscopy. So basically, you use the the light microscopy to identify where your peptides are interacting with your cells. And you do it in different time points. And once you identify this, and you do this all in cryogenic conditions, you go to another microscope that uses a laser that mills, so it cuts a very, very thin slice exactly where you identify the interaction. You take this thin slice and you go with this to the electron microscope, and then you look at the sample at basically atomic resolution. So you can get less than 10 angstrom resolution from a process like this. So it's called CryoEM. It's an emerging method. It hasn't been used for this particular technology, but it was used to see how viruses attack cells and organisms within the cells. And you know, we were talking before in a meeting about infrastructures in different universities. So also in Israel, we don't have this technology. And this is what I'm doing in my lab in Germany, because this CryoEM is emerging, but it's very, very expensive microscopes. And the setup, think that you have to have 3 different microscopes working on the same grids at the same time, and they all have to be really high end. So it's really difficult. But, you know, this is what we want to do, right? And obviously, naturally, something that we want to do is to use our mechanistic and structural understanding to find inhibitors of the virulence factors to be used in antivirulence therapeutics that might induce less microbial resistance. Because if you think about it, if you're trying to kill a microbe, it will fast try to find resistant mechanism. But if you're attacking the weapons of the microbes, the ones that make the infections more aggressive, maybe the resistance will not develop that fast. So you don't kill the microbe, but you help the body fight the microbial infection. And up to now, we found some inhibitors for the biofilm, but we haven't found anything for the PSMs in the staph aureus. We found maybe 1 compound, but it's too toxic, and other compounds, guess what it is? It's EGCG from green tea. So EGCG from green tea was shown beneficial also for Alzheimer's proteins, but it's apparently working also for bacteria. So now I have a collaborator, she drinks green tea every day, and she's 66 and she looks less than 40. So I'm trying, so in conferences of Alzheimer's, like people talking about drugs, but at the end people say, okay, we have to eat curry with curcumin, we have to drink green tea, and probably this is the best we can do to prevent Alzheimer's. And from the other direction, of course, we want to design new antimicrobials that will be sturdy, that will be tunable for biomedical and technological application. So, and I think that finding these inhibitors will also support the structure function studies, because if you find something that is working, then you know that what you defined is correct. So it's all like a circle that we want to achieve at the end. And if we aspire further, the place that I'm working in Germany has a biosafety level 3 of all microscopes. So what we are next trying to pursue is actually determining the structures directly from live biofilm. So to take the implants, like implants of patients that had infections, take the biofilm from that and trying to determine the structures directly from the samples. And if I really you know, thinking long term, I want to take brain samples of people that had both neurodegeneration and severe infections and looking what happens in these brains. And I can tell you, it's not published information, it's not for distribution, but collaborators from the medical hospital in Hamburg told me that the brains of people that died from corona, even if they didn't have Alzheimer's, they had some tau tangles. And people with Alzheimer's and corona, the entire plaque is changing. So even corona is having effects on Alzheimer's brains and neurodegeneration. And I think it's very important to pursue this direction about the connection between, you know, all the host pathogens that are related to microbes. So if we think about implication, of course, it's new antimicrobial drugs and materials for novel technology because we use the stability. I'm pretty sure that microbes communicate between themselves via the amyloids. We know that they form mixed biofilms. They can compensate even. It can be destructive interactions or synergistic interactions, and the host microbe interaction, everything that has to do with antimicrobial theory of the physiological role of human amyloids, how infection in the brain is leading to Alzheimer's, how microbial amyloids can template like mad cow disease, like the human amyloids. All of these theories need to be explored in depth from the molecular level to the in vivo settings. And it's time to say thank you. So all of the work, most of the work that I showed you was done by my Israeli lab. We are a small group. We have nice weather like you. When I'm giving this talk in Europe, I tell them, okay, this picture was taken in November, December, so you can be envious, but here you have the same. The beamlines that I'm using, the synchrotron, it's mostly at Grenoble at France, but I belong to EMBL, so I'm using the EMBL beamlines, and I'm a visiting professor at EMBL, so I have access to all their facility, which is tremendously helpful, especially in Corona. So my students couldn't travel, but because I'm there, I could travel during Corona. I traveled almost every month to use the synchrotron, and I could use the beamlines, which was very, very, very beneficial. We didn't have to stop. Robert is my postdoc in Germany. He was the one determining the structure together with Kai Grunewald. We got funding from the city of Hamburg to do this. You have to find money from different resources, people that will agree to host you on the infrastructure. You really have to find patches when you're looking for state-of-the-art methodology. And so Robert was solving the structure, and Fabio is a new postdoc that is now working for me also in Hamburg. And thank you for the funders, and thank you for your attention and being here.
No sé si tiene alguna pregunta o comentario para la doctora. Ingrid.
Thank you so much. This is fascinating. I have two questions. One is, how do amyloids reach the brain? I probably it's because I don't know about how prions work, but how do you imagine them crossing like from the gut or eventually all the way to the brain? And is it like through poking the membrane as you were showing? Like, do they trespass it, or are there transporters? And then the second question is that in the brain, do you imagine then that there is normally like cross alpha amyloid structures like serving an immune function, and then something happens that it becomes something like a maladaptive response of these structures, and then they become these cross beta plaques? Or how are you imagining these?
Okay, so starting with the first question, nobody really knows. There are theories that they go by the lymphatic system. There are some theories about direct gut-brain, so they don't even go to the circulation. There are some routes that are going directly to the brain. People thought about what you said, that maybe the blood-brain barrier is getting a little bit porous, and this is true when you have a severe infection, but it's very complicated. There was a recent paper coming out that compared the blood-brain barrier of Alzheimer's, normal people, and it was another disease. I don't remember what was the third disease, but actually in the Alzheimer's, the BBB was less penetratable, which is super, super weird. It's not what you expect. And how the people found chlamydia and herpes in the brain, so they have to penetrate somehow. Nobody knows. Some people think that there are some amyloid beta and alpha synuclein that they are entering the periphery, getting template there and go back. So there are so many hypotheses, and the answer is that nobody knows. Everything is open. And about the cross alpha, so amyloid beta and alpha synuclein, they are unstructured proteins. But people say, like now the common theory is that the oligomeric form and not the fibril form is the toxic one. And actually, amyloid beta and alpha-synuclein, they are helical in their oligomeric state. So maybe the helical configuration is encoding toxicity, and this is what we see. We see that the switch to helicity is correlated with toxicity. But to be honest, I also think that we shouldn't think about amyloid toxic entities. We should think about amyloid toxic process, because what we think from our peptides is that the formation of the fibril is happening on the membrane. So it's not a particular species that is toxic. It's the process that is toxic, and the membrane is playing a part. So it depends if you have the propensity to have these helical fibrils or not, or the helical oligomers. But if we see that, for example, if we give them heat shock and we stabilize them at beta form, so if you raise the temperature, they will normally go to beta. Not all of them, but most of them, they lose toxicity. So my imagination is that you have these species with all kind of morphologies, and on the membrane they assume what they assume.
Thank you so much. Fascinating.
¿Algún otro comentario? Sí,
It's mainly about what you were just saying about the energetics of the transition to forms of the amyloid. So do you know, it seems that the more stable form is like the helical one.
Beta.
The beta is the more stable one. Mm-hmm. Okay, so this transition, when it comes in contact with the lipid, do you think it's a stoichiometric process, or is just like a cascade process that some part of the sheet absorbs a lipid and then?
I imagine it more like a cascade. I call it a domino. Like in my brain, it's like a domino a little bit. Like even so on the membrane lipids, they're a little bit similar to detergents, and we know that salts, detergents, they're all stabilized helical conformations. Like this is known. Like many antimicrobial peptides, they're unstructured. They come with buffer with lipids, they turn helical. Yeah. So I think the chemistry, it's not stoichiometric necessarily. I don't think it's. I think that it's one lipid to a lot of peptides. And I think that even if you imagine that you already have a cross beta structure, which is really stable, but it's the same quaternary arrangement, then you have a lipid coming, and even one molecule is turning helical. It's like a domino effect. Tuk, 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 tuk. They change into the helical form, and they don't have to rearrange. They don't have to disassemble and assemble because it's the same quaternary arrangement. So it's like a domino. They switch their secondary structure and repack, but not assemble and disassemble. But I don't know. It's just my imagination, like thinking about this process. But energetically wise, the beta are much more stable. So the alpha ones, the alpha that stays alpha and never go into beta, they will dissolve in 60 degrees already. They will not stay stable. The one that can switch, that will switch into the beta, and then they will remain there with high temperature.
Okay. So how do you imagine that the lipid extraction from the membrane occurs, right? Because you have like this huge structure, but how can it just extract the lipids from the membrane?
What we see in confocal microscopy is that once you add the peptides, you have the cells, you add the peptides, and you see like pieces of the membranes around. So it's just a lytic process. But really small pieces of the membrane, and when you use dyes for the peptides and dyes for the membrane, they co-localize. So it seems that fibril aggregate and take some pieces of the membrane with them. So I think because the cross alpha has this hydrophobic core, it's recruiting the hydrophobic lipids into the core of the cross alpha. So it's just taking the lipids out. So basically rupturing the membrane, but not making a hole or a pore or something like this, just ripping it.
Yeah. Okay, so yeah, that sounds like very complicated, right? Because you have like the interaction with the hydrophilic part of the membrane, and then it has to interact with the hydrophobic. So
yes, But they're amphipathic helices, so they're supposed to be perfect for that because the surface of the fibril is actually hydrophobic and positively charged. So it's have these patches of hydrophobic and positive charges. So the positive, so they're all cationic peptides. So the cationic part is interacting with the negative parts of the membrane, and the hydrophobic part with the hydrophobic parts of the membrane.
Just one more question. So the amino acids that mediate the interaction between the alpha helixes and the are the same once the transition occurs, or?
I don't know. You mean the same ones that are on the surface?
Yeah, the ones that allow the beta. Ones that allow the
Not quite. So the one that we solve both structures for, in the alpha form, all the hydrophobics are in, and in the cross beta ones, it's less amphipathic. So the core of the fibril can also be a little bit hydrophilic, actually. So it does change the chemistry, and this change of chemistry of the surface of the fibril can be part of the explanation of why some are toxic and ones are not.
Yes, well, first, the presentation is so nice. Thank you. I want to ask you if, because in these infectious processes that you mentioned, there are a lot of immune mediators. How can they affect the amyloid structures?
It's a really good question. It's probably very relevant because in staph aureus infections, some of the lipoproteins, the ones that are carrying the HDL and the other one, the bad one, I forgot. So they are actually neutralizing some of these amyloids. So the body has mechanism from things that you don't even expect, like from lipid metabolism that are counteracting the amyloid. Maybe some of the proteins in our blood, like the albumin, are trying to fight them too. So there is constant battle between the human host and the pathogen of how to try to inhibit their amyloid formation. So some of these pathways are known.
Not. Dr. Landau, thank you very much for a nice talk.
Thank you.